we would just like to introduce our two wonderful speakers to everyone shortly before we get started. So Professor Tom Ellis is a professor of synthetic genome engineering at Imperial College London. He currently leads a team in doing a lot of awesome engineered living material research, and he previously completed his PhD at the University of Cambridge under Michael Waring, and then followed that up with some time in a biotech company before working with Jim Collins, one of the founding fathers of our field, and then returned to the UK to pursue further synthetic biology at the University of Cambridge before heading to Imperial. He is also a social media influencer on Twitter, as we can tell from his prolific tweets on this subject that I think many of us are familiar with and love. And I think Jose will introduce Professor Solomon.
Yes, indeed. Professor Kevin Salomon is an assistant professor of chemical and biomolecular engineering at the University of Delaware. He started studying a bachelor's degree in chemical engineering and bioengineering in McMaster University in Hamilton, Ontario. Then a chemical engineering practice and as a master's degree and a chemical engineering PhD, both from the MIT in Cambridge, Massachusetts. And finally, a chemical engineering position postdoc at the University of California, Santa Barbara. Right now, he's working on identifying and developing microbes from environmental communities well adapted for the deconstruction and upcycling of complex polymeric materials, according to his lab's website. And regarding synthetic biology, they are working on developing tools that can streamline organism engineering, accurately detect limits on biosensing in microbes required for precise control of key genes, enzymes, and metabolites. So welcome, Professor Salomon. Welcome, Professor Ellis. And Liche, I will send back the word to you.
Oh, thanks. And we are wondering if Professor Solomon and Professor Ellis, if you wouldn't mind just giving us a very short introduction about what you think about the current state of the field of synthetic biology and what the future may look like before we open it up to the floor for questions. Yeah.
Kevin, would you like to go first?
Let me get myself arranged here. Sorry. All right. Did something stupid. I will be back in a second. There we go. Actually, no, never mind, sorry. So yeah, I just prepared a handful of slides. So I just wanted to share some thoughts about where I see synthetic biology and where I see it heading. Now, before I kind of go into that, I just wanted to take a moment to do a quick promotion. I and Professor Nick Sandoval, we are guest editors for a journal called Synthetic and Systems Biotechnology. We have a special edition on engineering gene regulation, and so we're looking for papers pretty much for most things you think about when you think about synthetic biology, such as transcription factor engineering, using CRISPR-IA to control gene expression, cell-free systems, modeling of gene expression, et cetera. And so anybody in this room who has any interest in these topics, I would ask you to consider us and submit a contribution by the end of February next year. Now, so as you guys heard from my introduction, I did my PhD at MIT in the early 2000s, or the mid to late 2000s, and so I was there when synthetic biology was really coming to the forefront. The founders of Ginkgo were still students, and we all had regular meetings about the future. And so from those discussions and from my experiences, what I've really built my career about has been about really onboarding new chassis, looking and exploring novel systems that have a wide array of biosynthetic potential, so more potential than your typical E. coli or Saccharomyces. And what we found is that these systems, they have a number of unique challenges to their manipulation, which is why we're also developing foundational tools to engineer these organisms. And once we have these tools and organisms in place, we can think about how do we repurpose them? How do we use them to solve problems? And so naturally, you can imagine that my vision for the future is kind of going to revolve around these themes. So what do I think is synthetic biology? I think the 20th century or the birth of synthetic biology has been very good at focusing on genetics and how we can actually scale this up, right? So many of the seminal papers from Ellis, from Collins, and others have focused on how we can design genetic circuits for specific for specific purposes. What are the fundamental parts that we need to use to do these things, and how do we put them together? And that has also come with a number of foundational technologies. So for example, now DNA printing is very cheap and very scalable. We now have a wide, robust tool set to edit organisms. And to a first approximation, we even have computational tools that allow us to predict and design systems and even automate robots to build things just based on high-level specifications of function. And we're also beginning to see some burgeoning efforts in terms of genetic code expansion. And just from these foundational advances, I think there's been a wide, a diversity of applications that have arisen. So we can see lots of companies, and I apologize that this list is probably 4 or 5 years old. So there's many more companies that have come to the fore since then. But we see people thinking about meat substitutes. We see people looking about different types of materials such as engineered silk. We see sustainability in the form of renewable chemicals. We can see advances in biomedicine, such as, for example, Moderna, among other things, so the recent COVID vaccine. And to a first approximation, all of these innovations, they all arise from these foundational tools that I think were the core of 20th century synthetic biology. And so, oh, sorry, this is bad. What do I think? Sorry about that. What do I think is what do I think are the challenges of the 21st century? Well, I think for the 21st century, the common problems that I think synthetic biology will have to face are challenges in sustainability. So for example, as we have dwindling petrochemical reserves, as we have growing consumer waste, we need to think about ways of capturing and recycling carbon in a variety of different ways. And I think synthetic biology will play a role in this. Similarly, as the world gets wealthier, populations are growing and exploding, and there are ongoing questions about food security. And you can imagine that as populations grow and they need more food, but they also need more space to live. And so the food that we produce has to be produced in a smaller and smaller space. And so the question is, how do we improve nitrogen capture for crop production? How can we grow meat substitutes or protein substitutes that don't require as many resources? And similarly, as our population ages, how do we tackle the growing health concerns, such as, so for example, how do we replace failing organs? How do we develop the next generation monitoring systems that can spot a COVID pandemic in advance? How do we make treatments such as like these mRNA vaccines on the fly and rapidly? And so for the next 10 years, I think the first steps to getting to to tackling these challenges, I think, will focus on, for example, diversifying the chassis that we use and with an increasing focus on multicellular systems. So going away from single cellular E. coli, Saccharomyces to newer systems. And you can sort of see this now with, for example, new model strains such as Vibrio, people talking about organoids, people talking about microbial consortia. And I think as those technologies evolve and mature, we will see more and more complex systems to tackle these challenges. In a similar fashion, I think as we bring more new chassis online, we have to think about how do we engineer them. And so the gene editing technologies that we have all kind of evolved from the immune systems of bacteria. So starting with recombinant DNA technology with restriction enzymes, to engineered zinc finger nucleases and talons, and to the holy grail right now, which is CRISPR-Cas9 technology. However, one of the limitations of CRISPR-Cas9 is that there are these PAM sites that have to be present for where you program these enzymes to modify an organism. And what we're finding is that as we look at more non-model chassis, they have extreme GC biases, which make it very hard for traditional CRISPR to work. And so there's been a push for PAM-less CRISPR platforms, and there's been talk about using new emerging endonucleases such as prokaryotic argonauts that might, both of which don't use PAM and might be more flexible alternatives. And I think this growth in gene editing technologies will also lead to recoded organisms, more genetic code expansion, and really truly orthogonal systems for biosafety and biosecurity. And then, as one of the opening speakers, Professor Meyer mentioned, I think one of the underexploited facets of synthetic biology has been its potential for materials production. Biological molecules such as nucleic acids, proteins, they have exceptional design rules, and that lead to materials with a number of properties. We see, for example, with nucleic acids, people are making things such as DNA origami. So these are responsive DNA nanostructures that can be used for drug delivery. People are using proteins to create materials with different mechanical properties, such as, for example, spider silk. And these materials can be used to also template or lead to biohybrid materials such as nacre. And even looking at macromolecular assemblies such as viruses, these self-assembled protein nanostructures can be, again, complex with inorganic materials to make devices that go in our electronics, that go in our batteries, and so on. And so I think there is going to be a lot of opportunity here for synthetic biology making these biological materials. And so I guess just to wrap up, I think the immediate horizon, the questions that I think SynBio needs to answer are things such as scale. Yes, there's all this exciting potential for us to grow materials, to use interesting organisms in these large multicellular assemblages, but then what are the practical limits of scale? Like, how complex a multicellular culture can we engineer? If we could grow a house, how big a house could that be? How challenging would that be? And I think we need to kind of get some sense of what those ground rules are. And similarly, just like we've focused a lot on genetics in the last 20 years or so, I think there is, as we move on to mammalian systems and other more complex hosts, I think there is opportunities for new tools for genome engineering and also for epigenetics. And so that was my random rant, old man of the future. We can probe into it in detail. Thank you.
Thank you so much, Professor Solomon. That was actually a really great intro to both the past, present, and future of synthetic biology. Professor Ellis, I wasn't sure if you had some of your own opinions that you'd like to share at this juncture as well.
I wasn't expecting slides, so I've quickly made some on the fly as well. So
it's all good. We are a small group, intimate setting. Feel free.
So let's see if you can see mine. You see mine now? It's all shared? Great, yeah. So I mean, I echo, I believe a lot of what Kevin said is true. And kind of recently, we were lucky to be asked to write about the past 10 years in synthetic biology, which is kind of the second decade of synthetic biology. And that made us also look forward to what could possibly happen in the third decade of synthetic biology, which would be now. So I've had a chance over the last year or so to have some thoughts in this area, but we'll start here on this slide deck. I don't know if you can see the, yeah, you should be able to see that, right? The cursor. So this is from a review, I guess, from Jim Collins's lab quite a while ago. And they sort of put this timeline on that sort of shows from the beginning of synthetic biology in around 2000, when the first sort of research papers come out, what was achieved, what were the major achievements of that first 10 years. And I think, you know, enough time has gone by now that we can look back at this list and kind of reflect on it a bit. And it's interesting. I would say, you know, what's had the biggest impact, I would say, is, you know, SB1 meeting, first iGEM competition, you know, for actually having a community that came together, which is quite rare. Often when new sort of emergent topics occur in research, you know, they don't necessarily form into a coherent field very quickly. And they certainly don't form into one that has an active community that cares about sharing and cares about training in particular, right? Not every research group makes this their focus, but we were lucky in the field of synthetic biology that research groups all over the US and elsewhere in the world, but particularly groups based at MIT at the time, really cared about this and really thought early on, right, we need to train people in this area. iGEM might seem quite expensive these days as a competition to enter, but it has really driven a lot of the field that we've been able to train so many people through that route and copy some of the stuff done in iGEM, bring it back into universities as ways to train at universities or even to train at high schools. So there's, you know, we run at Imperial, you see behind me, we run like a mini iGEM as part of one of my teaching modules. You know, that sort of thing is very innovative and the kids love it. And you end up producing kids by the end of just one term or semester, as you say, that have really delved into synthetic biology in some way that now gives them confidence to design their own research projects or go pitch startup company ideas. And so it's really amazing. And that has driven a lot of what we see right now. The other thing, I guess, that sort of was a key success in those early years was the whole story around artemisinin and creating biofuels, all of that kind of metabolic engineering work that was being done and done well by people at both on the east coast of the US, like at MIT, but also particularly on the west coast around Jay Kiesling's group, with good stories about artemisinic acid and malaria, and also commercially interesting stories around biofuels and replacements for products in the petrochemical industry. Now we look back and particularly on biofuels, and obviously that sort of whole market and that idea changed. But those companies are still around, most of them, well, maybe a few of them, and still have good bottom lines, instead making specialty chemicals. The other thing I think that jumps out from this diagram is how the field thought a lot that the key successes were things like being able to do a logic gate or program a bacteria to do oscillations and things. And yes, scientifically, that's super cool. But 20 years on, right, like I don't see companies lining up to have an oscillator in their engineered product, right? So it's interesting to reflect back on that. Now, the second decade is kind of striking in that sort of once we hit that sort of 2010 moment, and particularly you can see as a landmark that the Venter Institute were able to make a fully synthetic genome, things started really kicking off, right? Like 2011, 2012. And that is, you know, you can look back at it and you think, why is that happening? Is that the standardization? Is that better methods? I personally think it's just, it takes like 5 years after you kick off something like iGEM before the rest of the world gets people who've done iGEM into their labs and starts doing stuff. So I think it took a while to go from training to amazing students who can lead these projects. Plus, I know from the UK side, it took a while as well to show the successes that were happening in small clusters in the US and convince the government and the research funders, like, you should fund that stuff here, and then we will get a cluster. But I mean, you just look at this list, the kind of stuff that we've done from a scientific point of view is kind of bonkers, right? Like E. coli engineered with 6 bases of DNA, entire synthetic chromosomes, whole cell simulations of an entire cell, genomic recoding, making gene circuits with de novo designed proteins by 2019, Hannah Samad and David Baker. We don't even stick CRISPR on this list, but there's a pretty strong claim that a lot of the early advances in CRISPR came from the synthetic biology community, right? Like after Doudna, the first 2 papers that reprogrammed CRISPR to do other things were one from George Church's lab, where everyone was heavily invested with trying to find ways to in vivo edit E. coli to change its codons. And the other was from a guy at MIT who had been a fellow in George Church's lab the year before, right, Fen Zhang, who had been on the project about how can you do in vivo editing. And that's just CRISPR editing. CRISPR as a protein that you can program to go places, like the DCAS9, that was Wendell Lim, Adam Arkin, Jennifer Doudna, and Jonathan Weisman's groups coming together. So that was really a synthetic biology product as well. So this second decade has been really impressive, and has led to clearly, many big groups, small groups all over the world being able to achieve all sorts of things. We've had at least 3 or 4 synthetic genomes complete now, and published, and updated versions and upgrades, and those are genomes. We have automated design of logic circuits. It's pretty cool, and then you put it the other way, and it's like there are thousands of people doing iGen and other things like iGen. There's training courses at universities everywhere. And now there's also, an abundance of jobs in industry. And that industry has been created, at least half of it, by people who were doing synthetic biology in research groups earlier. And how many new synthetic biology startups and companies appear each year? It's like dozens, if not hundreds. So it's pretty impressive. So where do we go from here, right? Now that we've taken over the world. So I was asked this for a meeting about a year ago to list like 10 things for the future. And a couple of undergraduate students in my group and I wrote these up recently and sent it to a journal called Engineering Biology, which is a bit of a hidden journal that not many people might know about. But if you go to my group website or Google Scholar, you can find the link to an article discussing these. So what is needed in the future? So these first ones are kind of ones we know are happening anyway. Industrialization and automation, right? So lots of research groups, foundries are doing this, but actually where the real progress probably is happening is at companies, particularly ones that follow the same model that Ginkgo Bioworks or Synthase or Opentrons are now going to do, or Zymogen, right? Where there's a lot of industrial process, automation of the design, build, test, learn cycle, and particularly focusing on strain optimization for strains that make molecules, like basically metabolic engineering. Machine learning for DNA design, right? So machine learning's everywhere. You can't get away from it now. It's not going to go away. And I think that there's such a huge opportunity for it in synthetic biology because fundamentally DNA is code that is written in a language, like certain strings of letters are understood by the cell in certain orders and context to mean things and direct the cell to do stuff, right? And we don't yet have full understanding of this language and what this code is because we've only started learning it in the last 70, 80 years in molecular biology and biology. But we're getting more and more information on this. And that's kind of a very machine learning relevant problem, right? Like learning how to write a language, having just been given a ton of data, is something that we can see like Google Translate can easily do. So Google Translate could probably be used at some point to start translating DNA and telling us what function it might have, although it's obviously clearly a larger problem than just straight language translation. Synthetic cells from biochemistry is a big area. There's multiple communities out there that are partly in synthetic biology and sometimes just a bit more adjacent to synthetic biology, looking to put together biochemical reactions into things like liposomes and vesicles and try to build a cell, and that's the name of the US community, or make a synthetic cell, which is more the name of the sort of European community. And that's kind of exciting that we might be able to put a whole load of components into a vesicle and it start to actually perform like life, and then maybe we can stick DNA programs into it and they can become self-sustaining. The next one, okay, no, this is a joke. It's not a real one, but it would be fun. But let's not everyone aim for this. But I guess one thing I kind of like about this as a joke is that I think consumer products from synthetic biology are likely to happen at some point. and I would encourage them to happen, because GM is an interesting thing. People are not so keen on like the local fields near their beauty spot being full of a GM crop, but that's a completely different consideration to oh, I really like this fun thing and I want to have it in my house, right? That's a personal decision. Whereas we look around and see like GM crops as something that's in nature, which is something that is shared up by all of us who live around that. But, you know, taking a GM medicine or having a GM product in your house is a personal choice, and many people will be for it and see benefit from it. So these are the other ones. So that one didn't really make the top 10, right? So cellular communities and multicellularity, it's kind of obvious, right? Like we've done really well at single cell level, and particularly in microbes and synthetic biology. We've got to get better at understanding this entire new dimension where you can take complexity and you can take functionality, which is to include multiple different cells that do different tasks and have themselves arranged into multicellular systems. Designing with whole cell simulations I find quite exciting. We have a whole cell simulation of a cell cycle for Mycoplasma genitalium. There's one for E. coli coming, I think. There's one for a human cell in progress. How do we design, build, test, learn with the ability to put our designs into a simulator, as opposed to just having them as done as standalone models and thinking about them in that respect. And then the bottom one, I think this is a really big one and a very broad one, which Kevin already touched, right, is sustainability goals. I mean, the United Nations has multiple different sustainability goals. And if you look at that list of those, it's pretty hard to find any of those goals that don't involve biology, because they're things to do with healthcare, water purity, scarcity of resources around food, right? And so all these things link in with biology. And I think the younger generation, people younger than me who have more hair than me, probably are growing up knowing there's going to be this crisis happening, and it's going to get worse for their lifetimes, not just mine. And I think there's going to be a big push for anything in sustainability. I should add as well, if you're listening to this, and you're thinking where next, right? A synthetic biology company whose product helps against the climate crisis and is something to do with sustainability, that's like an open goal, right? Like venture capital will fund you because you're a synthetic biology startup company, and it's just crazy right now. But beyond that, venture capital will just go nuts and throw money at you if it's to do with sustainability, because there's a lot of money that institutions, banks, and everyone wants to invest in this area, both for political reasons, but also because the returns can be really, really good if sustainability is enforced and is taken up as much as we think it needs to be. Two more areas, right? So living materials. These are two that obviously my research group work on. I want to get to the point, not just us, but our field, where we can design at the DNA level DNA circuits that don't just make a bacteria oscillate and do GFP, but that define a material that gets made in terms of its dimensions, its shape, and its material properties, right? That's a real grand challenge of being able to get cells to work together, differentiate, make multiple things, and make them in shapes and structures. And then custom genomes. If we can do synthetic genomes and we can recode those, let's do something even more out there. Like let's design entire at the genome level in the future for a cell that does a specific task, or a genome that changes over time so that cells change their tasks over time. Final two ones, and these are big ones, really ones I think I would like people to think about. Biosurveillance. I think biology has the power to detect anything anywhere, really. If you think about what we want to detect, it's normally interfaced with biology in some fashion, right? So the soil, in our bodies, on our skin, on the side of, on the seat in an airplane or whatever, right? We can do biosensors, but think about like if we could really do this really well and we could find ways to have cells detecting what's around them, or molecules detecting what they're bumping into, and quickly transmit that information to us and get it on the web or email it to us or something, right? It would be amazing if we could harness the power of the fact that biology is sensing everything all around it all the time, and be able to plug into that and get that information. It would be revolutionary. And then the final one, I think, is to have more of an understanding of how to control and harness evolution, because the synthetic biology field is good at writing DNA programs that will go into E. coli and do a certain thing for a short period of time. But we need to start thinking of the dimension of time itself, that once DNA goes into an organism, selective pressures act upon it, and it mutates, it will get selected for and against, right? And so if we somehow we can get more predictive or have more control over that, then we could really go somewhere. Like, you could put really badly designed things into cells and design them in a way that they self-optimize over time, really accelerate our engineering. So there you go. Those are my thoughts. Welcome to my mind and that of my students. So I'll stop sharing now. Maybe we can get on with some Q&A.
Awesome. Yeah, thank you so much, Professor Ellis. That was a very nice, concise summary of a lot of the SynBio ideas that have been bouncing around our heads and the fields over the past 20 years too. Great. And yeah, I guess it would be nice for us to move on to the Q&A portion of this panel discussion right now. We'll be taking questions. You can either unmute yourselves, or you can feel free to drop a question in the chat either way, and we'll let the speakers answer them. Does anyone have any questions about the general future of synthetic biology, or any points about the past? Oh, I think Shyam has a question. Shyam, we can't hear you. Sorry. Okay.
How about now?
Yeah, we can hear you now.
Oh wait, now I don't.
Or you can type your question in the chat. If you... Okay.
There we go. On your last point about on evolution, what more? I guess dynamic control of evolution. What more would we need to think about beyond things like, I guess, the repetitive DNA, or I guess things that, say, the evolutionary failure mode calculator from the Barrick lab might be able to predict?
I think, yeah, I guess we're being quite one-dimensional even there, right? Like, your evolutionary failure mode calculator is like, okay, if this is repetitive DNA and it's a stress to the cell, so it causes a burden, the repetitive DNA, it will get deleted at some point, and then you'll grow, and because your thing is stressful. Like, we already, I think Guibert Stan's group, which is the group next door to mine, they already have, like, they've used evolutionary failure mode plus the work we and Guy's done on burden, combine the two, and you can already predict, like, failure rates for certain things and that evolution. But you've got to think more dynamic here. We want to be putting organisms into bioreactors. We want to be putting organisms into dynamic systems, and that those selective pressures are going to change over time, right? And it'd be nice to, so Gorachowski and others are thinking, well, how can you design for that? How can you have that in your design? But you also want to have control, and ideally dynamic control. So you could think about having things like DNA editing online in a cell, so that over time, you can specifically mutate certain regions or repair things as they evolve. I think there's a huge opportunity for sort of dynamic CRISPR at the moment that people aren't taking opportunity of, and using kind of like having a cell have the machinery inside and having it turn on and on at different times. Others are doing things like directed evolution, continual in vivo systems. And so that's a really great start, that you could think about directing those to certain regions at certain times to improve something.
So with respect to biomanufacturing, this is the same thing as like the cheater problem, right?
Yeah, that's one way to think about it, right? And I guess you might also say the other half of this story is how do you incentivize a cell to keep a DNA program that makes it do something that isn't just replication, right? And so there's some smart ways to think about that. It's a difficult one, right? Because it just wants to grow and divide.
And it's also difficult because like to incentivize the cell for a specific program, that's very program specific too, right?
Yeah.
Awesome. Thanks, Shyam. Okay, I think we have two questions now. I think Professor.
I think this is an interesting.
Oh, sorry. No.
Sorry, I'm just gonna say that I think Professor Ellis brought up some really interesting points for how we need to tackle this. I guess the other strategy, which is the strategy that I prefer, is to find chassis that are optimized for this in the first place, so that the selective pressures are they're not zero, but they're certainly less intense, which also comes with its own challenges, because there's a reason why we've been avoiding those organisms, because they're not as genetically tractable as, say, for example, things that we're familiar with.
Yeah, I have to say, one thing that stuck in my mind when I was on sabbatical in 2018, I visited Adam Arkin for the afternoon, and he said to me, like, it's amazing how good we are at engineering E. coli to do some really, really fancy stuff, but I can go out in nature and find some other organism or community of organisms and just do one dumb mutation to it, and it will outperform E. coli in whatever task you give me.
Yeah, nature has been really good at solving a lot of the problems that we think are new and interesting.
Yeah.
So may I comment a little bit, because this is actually the most interesting topic, entire my life. I mean, the evolution, because that is my actually job talk idea about how to fight against the evolution as a bioengineer. And actually, that is my NSF career all about the evolution, and also burden calculator was in my NSF career, but unfortunately, I was scooped by you, Tom and Gagi, and also scooped by my own colleague, Fu Zhongjang, and because I share my idea with him, and then unfortunately I scooped by him. But that's fine. But but still, this is a very challenging problem, as a bioengineer, because I mean this is a continuous battle between us and our engineered bacteria, because bacteria not that much favor working for us. They just want to survive. They just want to live. I mean, just divide, and that's the only goal they have. Our goal is to make them working for us. So as you can imagine, this is a very challenging problem, and then that's why I believe the Tom and Guy developed the burden calculator. I love that paper, by the way, and unfortunately we are scooped, but I love that paper anyway. And then, but still, genetic circuit, that is prone to, mutation. And then another favorite paper is published in, I think, the Journal of Biological Engineering by some UFW people at the time, failure mode. I mean, just they kind of use some, evolutionary kind of experiment, and then they show kind of the very nice kind of failure. And that's my key paper when I talk about my job idea 10 years ago, and that's the foundation of my work. But my point here is, I mean, there are some methods we could address the problem. That one of them, I believe, is the redundancy. So nature has a redundant copy of the gene. So one break, the other kick in, and then do something, the same thing. And then also there are some other way we could use, including typically we use the plasmid. The plasmid tend to be roast, but another way we people the nature actually use, also people already use, is the plasmid addiction system. In that way, essential gene is in the plasmid, and then we knock out the whatever essential gene, and then now bacteria need to keep those plasmid, otherwise there is no survivor. And then in that way we could also remove entirely the antibiotic resistant gene in the plasmid. That's very important in terms of biocontainment perspective.
I think on the whole, though, I mean, you're talking about fighting, and these guys don't want to work with us or whatever. You got to think, like, you sound like a Roman emperor complaining about slaves, right? And
right.
We know from past history that, like, you have to get away from that. As you have to develop democracy, you have to have you have to have the minions want to work for you or whatever, right?
That's exactly my point. So
it's more than just these sort of little changes. I think if we're going to really be able to harness evolution, we have to kind of somehow have a real rethink about what we're doing. And I think Kevin's right in that, like, other organisms, right? Like making E. coli do all this other stuff. It's not happy doing it, right? Because
right, exactly. So that's
why it's not used to it. But you can go pick out other organisms which are probably going to be happier to evolve and to work to do this. And I think one thing that we're really not doing is looking at the other, the non-microbial biology, where a tree will have cells do stuff that is, an insane amount of the budget of that cell, right? Like to make a flower or to make a seed or something. Whereas the other rest of it, right? So you got to think about multicellularity as well. Like, okay, have cells that grow, divide, and do the replication, but have other cells that make the stuff. Did someone in the chat have a multicellular question?
Right. So, but actually, that's exactly my point I wanted to make. My second point was actually talking about the Kevin's point. So that's another reason I actually start my lab, basically focusing on the non-model organism, because I believe the E. coli is good and easy to engineer, but at the end of the day, real application require non-model organisms. So entire my lab actually, when I started, basically focused on how to engineer non-model organism, how to develop tools, and that's basically the half of my lab actually focused on since the beginning of my lab. And you point out very nice thing in the emperor slave thing, and I completely agree with you, because I believe many problems, including climate crisis, that is because we as humans think the planet, and we are not owner of the planet. The planet is to belong to everyone, not only human, but also animal or insect, even plants. This is to belong to everyone, not just human. But we, many decades, many centuries, people think we own the planet, and even people thinking about or talking about land use, and then kind of improve the efficiency of the photosynthetic bacteria. That is kind of good thing for the synthetic biologists, but to me this is kind of nonsense, because we do not own our planet. We just follow the planet, just living here. So we need to completely change our mindset. Otherwise, there is no solution for the climate crisis. And then we need to completely change the mindset. That's my point. Actually, that's why I'm working on the non-model organism, because I wanted to minimize any genetic modification, because that we should live together with our bacteria.
I absolutely agree, Professor Moon. But I think in the interest of time, we might move on, because I think I see another question in the chat, if that's okay. And this is really a question about moving towards multicellular systems, and whether our speakers had any thoughts on recombinant proteins that encode mammalian post-translational modifications that are often very beneficial to us, such as human insulin. I don't know if Professor Solomon and Professor Ellis, if you might have some thoughts.
I mean, I don't have anything specific, but I mean, yes, there is a need for those types of technologies. And so, for example, the work from Matt Ellis, among others, I think has been exceptional in this space in getting E. coli to add post-translational modifications. Although I think there's also opportunities to have yeast, so Saccharomyces, things that, again, more naturally make these things do it more efficiently than trying to make E. coli do it for 2 days, after which point it breaks and never does it again. Sorry, I'm like I did my PhD in E. coli. I love E. coli, but what I learned from my PhD is just because we can do something in E. coli doesn't mean we should. and so I purposefully poo-poo it at every opportunity, although there's a lot of cool things we can do in E. coli.
I don't have anything to add to that. So maybe one of the people with a hand up can come in now and ask another question.
Yeah. Let's assign the word to Karilaos Ioannidis.
Yep. Thank you very much. So my question is about something that I often see not given much attention to in thoughts about what the future of synthetic biology is. And that is specifically synthetic biology as a framework or method to uncover new biology. And I think that what those early generation circuits in the first decade did was they taught us so much about the rich repertoire of behaviors that transcriptional circuits can endow cells with. And my question now has 2 fronts. Consider that transcription, translation, and metabolism is the processes that synthetic biology has mostly focused on. What other kind of cellular process would be the next target, and how much could we learn by trying to engineer it? And the second one is what aspects of multicellularity, for example, how much do you think that a bottom-up synthetic biology approach will be able to delineate design principles for multicellular behaviors or microbial consortia?
Professor Solomon or Professor Ellis, would you like to answer?
I think for the latter question about design principles for multicellularity and synthetic consortia, it's quite difficult from what I've seen. There are groups making good progress on this. So in London near me, there's Chris Barnes's group who's doing some very good stuff on the theoretical framework for consortia and multicellularity. And I think in the US, Ophelia Venturi, I think that's her name, in the Midwest is doing great work in that space as well. I don't know, have you ever heard of 3-body problem, 2-body problem, right? If you have 2 things that have to interact and have feedback on one another, you can kind of predict it. But once you get to 3, it starts to get, you literally head into chaos theory, right? And so that's something that means that it's going to be pretty hard to make headway in this space as we get more and more complex interactions. The first question, I think, was learning from biology. So I certainly think we can learn from biology by trying to learn how you make a cell consortia, considering in theory it should be chaotic very quickly. So I think there's people working on doing that from a synthetic bottom-up point of view are really doing some interesting things that are going to help give us fundamental understanding of how things like a microbiome or a holobiont come into force and how they evolve. And so I certainly think that's an area. Kevin?
Sorry, just to clarify, was that the question that synthetic biology can learn from biology, or was it the other way around, that biologists could apply and use synthetic biology?
It was the other way around, actually, how we can learn new biology by doing synthetic biology.
Yeah, okay. And so my answer to that is I think people already are. I think this is I think synthetic biology in this regard is a victim of its success, in that I think lots of the foundational approaches for how we think and conduct synthetic biology have just become routine now. And so in a way, all biologists kind of use it without actively thinking of it in that way. Because I mean, certainly I think as Professor Ellis kind of mentioned, building things from the bottom up, synthetically creating artificial gene circuits to kind of recreate the principles that one hypothesizes are responsible for a given phenotype. I think that has, in fact, become a standard for how we do it, how biologists work. Although I guess a proper biologist will probably may not 100% agree with me, but that from my perspective, that's what it looks like they're doing to me.
Thank you both for your answers. Yeah, and I think we have another question, another hand raised from Alex did so, if you'd like to unmute yourself and speak.
Yeah, so one thing you both mentioned in your presentation was using machine learning and AI for predicting systems and using that for design. I wonder how you think that would affect how we design systems, because right now there's kind of, we can do rational design of systems or we can do directed evolution type things. But with machine learning, it's kind of rational design, but kind of a black box where we put in what we want and kind of get out something like predictions and try and work with it. And it seems like it would significantly affect the design process of these biological systems. And especially with things like AlphaFold and stuff like that, we can see that there are lots of new opportunities for things like that opening up. So I just wonder, how do you guys think that would affect the design process in synthetic biology?
Quickly, I think to some extent it takes away, not all, but a large amount of the trial error and directed evolution, for example, like applying machine learning first before doing directed evolution experiment on an enzyme design or something, making use of AlphaFold, will give you a big head start. But at some point, probably you need to also then do directed evolution, and the data you would get back from that, because directed evolution is normally done with thousands or millions of cells, can feed back onto that as well. So that's one aspect of it, that it just sort of can become part of the pipeline. And the other area where I think it is I kind of have the feeling that it's going to be replacing quite a lot of what we've done with sort of modeling and simulation to some extent, in that it's just basically going to say, well, look, previously we sat down with a pen and paper and thought very hard about equations, but let's just let data come back on this. But it is a big it's definitely something we need to be mindful of, that if we just go down the machine learning route and just hand over large parts of the design process to it, are we really going to, we might make a lot of short-term gains, but then we might put ourselves into a sort of a bottleneck of development of the field, in that we've lost sort of an idea of how it should really be done because we've just handed it all over to black boxes.
I want to echo that. I think for areas where our understanding is fairly mature and refined, I think there's a lot of short-term and even long-term gains that are possible with machine learning and AI. However, for things such as, for example, multicellularity, I think those systems are too complex to distill it down to an empirical design rules, because I think those things, because they're bordered on chaotic, there are multiple regimes that you can access. And if you don't have data from that entire design space, you will underpredict and lock yourself into scenarios that may or may not be feasible once you go out in the real world. And without any, because you've black boxed it, you wouldn't be able to recognize or solve it. So I do think that there's still room for human intervention to build robust models and design these systems.
Thank you both. I wasn't sure if we had more questions. I think Jose himself, I think, had a question as well.
Yes, but I just wanted to give some time in case some member of the audience wanted to ask something. But Professor Solomon, Professor Ellis, what is your vision to a global endeavor for synthetic biology? How do you see, or what are the opportunities that you see that can be taken to make the field globally accessible, especially for countries that are in their way to achieve scientific and technological proficiency?
Good question. So I guess there's two ways of thinking about it, right? So one way you could get is kind of moonshot style, right? Like some people brainstorm an idea for an amazing project that the community should all do together, and the whole world can come together and feel part of it, right? But it's very hard to do that, because if you for a start, moonshot projects are often done in competition, right? Like flying, who can get to the moon quickest, who can sequence the human genome quickest. That was a competition. Who can develop nuclear weapons the quickest, right? So it's not necessarily a great thing to do as the world. It tends to have been done as a race between superpowers. And I think also the other problem with a moonshot is you can choose one project, but loads of people might disagree with you, and then you change your mind. And that's kind of happened for people like George Church, who proposed synthesis of a human genome, and then people were like, well, what would that be useful for? And then it became a big debate, and it didn't really go anywhere. So I think it's better that the community just find ways to work together more and more, to train together, to train to similar standards more and more around the world, and to maybe identify lots of little goals. And I think I've seen the US have really recently, whereas, other countries like we had in the UK a long time ago, nearly 10 years ago, we did a roadmap for what we want synthetic biology to achieve in the UK over the 10-year period. But more recently, the Engineering Biology Research Council in the US produced a much more detailed technical roadmap of achievements that they want the community to achieve over 10, 20, and 30 years, with milestones and everything. I think that sort of thing expanded to building from that, letting the whole world in on that, and expanding it to include training, and particularly anything to do with climate and sustainability, is really the way forward. It sounds quite boring. It's like committees and goals and targets, but that's how the real world works, really.
I mean, I definitely think synthetic biology can be democratized, and it is accessible to the entire planet. I think the challenge is that as we build increasingly complex systems, there are too many unknowns. And so the way that we address those is through trial and error. And so, for example, we benefit from economies of scale in terms of DNA synthesis, among other things. And that infrastructure, I think, is less accessible. And so while I think we can definitely think about how to democratize synthetic biology and have it be applied worldwide in terms of pushing forward in terms of these moonshot programs. I think, at least for now, I don't see quite a path forward to making it available to all, just because of the infrastructure that's involved and to keep up with the pace that we currently have.
Thank you. Liche, we have
I guess we only have 2 minutes
questions.
left. I don't know if the speakers would mind taking one more question, or possibly two, just that we have in the chat.
Or before we do that, I just wanted to address the E. coli discussion. I poo-pooed E. coli a lot. By all means, I think a lot of the fundamental synthetic biology that we've done has relied on the tractability of synthetic of E. coli. It allows us to, as was mentioned, to develop a proof of principle. But then I think as we think about actually solving real problems, E. coli as a chassis might not be the optimal platform. And so the question then becomes, how do we take the approaches and technologies that we've learned from E. coli and adapt them to a new host, which unfortunately is non-trivial. It's not a one-to-one transference, which I think is underappreciated by the community, at least.
Thanks for addressing that. For sure. So I think one of the questions that we had was about what types of advancements are required to design microorganisms for deployment in space. I know the hour is up, so speakers, if you have to leave, we completely understand, but if you'd like to stick around to answer this last question, or perhaps provide your contact email where people may be able to reach you for further questions, that's also something we could arrange. I wasn't sure what your availability is like.
I'm gonna have to go because my wife wants me to make dinner. It's the
No
evening.
problem. Thank you so
Lovely
much.
talking. I'll go and think about space. You guys can talk about it.
No
short
problem.
answer to that one. So EBRC thinking about road mapping, about the space application, and also currently DARPA, has call regarding space basically engineering using synthetic biology. And then actually I attended that proposal meeting myself, and also I present there. And then now even DARPA thinking about, so that, as soon as DARPA thinking about something, that means kind of that is the next 10-year thing. So
it is, and so like to answer the question about what's required, I think for the first approximation, I think lots of what we know about synthetic biology applies. I think the challenge here becomes in the life support systems, because in space you have finite resources, and so the question is, how do you recycle dead cells? And so, and that I think requires a more sophisticated appreciation of ecology, and again, multicellular systems where you have things that can recycle dead cells, recapture those nutrients, so that you have something that's self-sustaining, as opposed to what you do in your lab where you make LB every other day and kind of throw it in there. And then once it's done, you feed them again. In space, we don't have that luxury of bringing all that stuff with us. So how do you
think about even you recycle your own urine?
Yeah.
So that's kind of actually happening in the space, and then that is also very relevant to the current situation in this planet about the climate crisis. So all the problem now talking about in the DARPA and then NASA, that is applicable to current solution toward sustainable development or the climate crisis solving, because that's why DARPA thinking about this big problem, and then that's why people kind of moving into that area.
Thank you, Professor Moon. I do think we are a couple of minutes outside of our originally allocated time, so we might have to end the session here. But thank you so much, everyone, for coming. And yeah, I hope you have a good rest of the day and enjoy the remaining 2 hours of Gogec at our award ceremony. Take care, everyone.